The cells, the ultimate consumers of the materials delivered to them by the blood, have some degree of control over their own supply. Tissues that are active and demand more in nutrient and oxygen delivery can open up the spigots, so to speak, to allow more blood to flow in. Conversely, cells that have lower metabolic demands can ramp down their own blood supply to match supply with demand. This kind of local control of blood flow is known as metabolic autoregulation. It works something like this. Imagine you have a collection of cells and they're consuming oxygen at a certain molar rate, m dot sub O2, m. Nearby, there's a blood supply that can deliver oxygen at a molar rate, m dot sub O2, Bl, which of course is carried in on a certain volume flow rate of blood. Thus, we have an oxygen consumption rate and an oxygen delivery rate. In a well-regulated system, the oxygen delivery rate will equal the consumption rate, that is, supply will equal demand. If cells are to regulate their own oxygen delivery rate, there has to be some way for the cells to control the blood delivery rate. The question, of course, is how do they do that? Cells don't have any direct way of conveying information to the vessel delivering the blood. They don't have nerves or things like that, so how do they do it? What they appear to do is use proxies that can indicate to the blood vessel how closely delivery matches demand. Probably the most significant proxy is oxygen concentration itself. The local oxygen concentration is going to depend upon the balance between the molar delivery rate and the molar consumption rate. Molar delivery is, of course, a function of blood flow delivery, while molar consumption rate is the function of local cell metabolism. When these two fluxes are approximately matched, the local oxygen concentration will be roughly steady with respect to time. If consumption rate exceeds delivery rate, local oxygen concentration will decline, and conversely, if the molar delivery rate is greater than the consumption rate, local oxygen concentration will increase. The ideal is for delivery rate to be matched exactly with consumption rate. If consumption exceeds delivery, the circulatory system is failing to do its job. If delivery exceeds consumption, the circulatory system is wasting energy to deliver blood and oxygen to cells that don't need it. How does this actually work in practice? Let's take a look at a model vascular bed, which we've seen already in another context. We have cells that are embedded in a matrix of capillaries, which are fed by arterioles and drained ultimately by venules. Blood can either flow through the capillaries or the blood can bypass the capillary bed altogether through an anastomosis. The arterioles and venules are invested with smooth muscle, and the tension of the smooth muscle wrapping can influence how much blood is delivered to a capillary bed and how much is drained away. As we've seen, this is under mostly endothelial control. Nitric oxide produced by the endothelial cells can produce a vasodilation, and endothelin can produce a vasoconstriction. Vascular smooth muscle tension in the arterioles and venules is also under a degree of top-down or nervous control. There's an additional level of control, though, and this occurs at the base of the capillaries that lead from the arterioles. These are the precapillary sphincters. They are not innervated at all, and the tension in those is controlled by local conditions. Let's see first how the precapillary sphincters work to channel the blood flow. When the precapillary sphincters are tensed, blood flow bypasses the capillary bed and either travels downstream to another bed or short-circuits the bed through the arteriovenous anastomosis. When the precapillary sphincter is relaxed, however, blood is diverted into the capillaries, where materials can be exchanged by diffusion with the cells that are embedded among the capillaries. By opening one precapillary sphincter and leaving the others closed, one can even adjust blood flow to certain regions within a capillary bed. How all this works is still an area of active research, but in some tissues, like the brain, it's pretty well worked out. The brain is a particularly easy case because those cells are very sensitive to variations of local oxygen concentration, and this makes for robust experiments to work out the details. It's also pretty well established that there's a great deal of fine local control of oxygen delivery within the brain. To illustrate this, here's a functional magnetic resonance image of the left and right hemispheres of the brain of a person suffering from so-called body dysmorphic disorder, which seems to be tied into distortions of people's own body image. Anorexia nervosa, in which someone thinks she is overweight even if she's objectively quite thin, is one example of a body dysmorphic disorder. Those details are less important, though, than what I want to illustrate with this image, which is that there's a great deal of variation of local metabolic rate within the brain. Areas of the brain that light up, so to speak, in an fMRI image like this are areas of the brain that are consuming oxygen at higher rates than others. That's what an fMRI measures, local oxygen consumption rate. These areas of local high oxygen consumption have to be supplied by local variations of high capillary blood flow. Regulation of blood flow in the brain is on demand. That is to say, if a local patch of brain tissue consumes oxygen at a certain rate, the circulatory system simply delivers it with priority over almost any other tissue in the body. And here's how it works. Imagine we have a local capillary bed with a brain cell consuming oxygen. If delivery by blood flow fails to meet demand, the local oxygen concentration will decline. The cell can sense this local oxygen decline and, in response, secretes nitric oxide. Nitric oxide is a gaseous neurotransmitter, and therefore it diffuses very quickly through the tissues. When it reaches the precapillary sphincters, it dilates them, allowing more blood and hence oxygen to flow into the capillaries. Thus, we have direct control by cells over the spigots that govern blood flow and oxygen delivery to themselves. The precapillary sphincters are, in essence, slaves to the metabolic demands of the local customers, the cells. Local control of blood flow in the brain is the prime example of a selfish regulatory system at work. Selfish regulation cannot be the only operating principle here, though, because, like all selfish systems, their success depends upon support by other systems. In this instance, selfish regulation of blood flow depends upon a reliable supply of blood upstream, that is, blood supplied with sufficient and reliable pressure so that blood flow can be adjusted simply by turning the spigots of the precapillary sphincters on or off. However, turning those spigots has inevitable impacts upstream, which places burdens on the upstream systems that supply the blood. Let's turn now to some of those impacts and their consequences.